welcome to our snow day edition of Molecules, Cells, and Genes. I've broken Lecture 6 in terms of its slides down into 4 different videos purely for the purposes of keeping our file sizes somewhat reasonable. But note that you're going to have to watch 6.1 through 6.4 in their entirety to be able to complete all of the lectures for this class. We do have a Socrative quiz, so go ahead and open up your Socrative student app and make sure to put in the room code. You're going to want to pace yourself while you do these. Pause the video as needed to give yourself time, and then you can resume the video again after you've answered the question. Don't forget, you're not getting graded on whether or not you're accurate, so use it as an opportunity to practice and test yourself. I'd also like to remind you that the file storage for our web page is limited, and I've given you a lot of videos that you can watch, the videos that you've seen in our PowerPoint slides, but I have to remove them periodically to make space for new videos, like today's lecture. Please remove any videos you want from lectures 2 through 4. You can either watch them or save them to be able to view later, but you have to do so by the end of today. You can find these videos in the course document section of the web page. So let's go ahead and get started. We're using our Socrative app to do a review. We're going to walk through step by step the creation of a protein and see how you do. My first question is, the chromatin within a nucleus contains genetic information encoding for proteins. The first step in the process of creating a functional protein product is the creation of an mRNA, also known as a messenger RNA copy. Remember our central dogma, DNA to RNA to protein. Remember that you have to go through this essential copying stage from one to the next. So what I'd like to know is, how do proteins facilitate the next step? Once you've created this messenger RNA, what happens? Nuclear pores are the proteins that facilitate the exit of this mRNA from the nucleus and into the cytoplasm. It's an interesting and critical step where the proteins that are later produced come back and facilitate these earlier steps. Don't forget, just because it goes DNA, RNA, protein doesn't mean everything that happens to the DNA is only DNA. We're going to have proteins that are involved in this process throughout its entirety. Proteins make up 50% of the dry mass of the cells. They're there all the time doing lots of important steps. This is just one example. So now that we've moved our messenger RNA into the cytoplasm, what enzyme is responsible for the next step? Ribosomes. Remember, ribosomes are enzymes, and they're made of both proteins, as you know proteins can be enzymes, as well as an RNA component. Ribosomes RNA is made inside of the nucleolus. We call this rRNA, kind of like how the little M denotes messenger for messenger RNA. We have R for ribosomal RNA. Both of these different macromolecules interact with one another to create a 3D shape that acts as the enzyme that we know as ribosomes. These ribosomes are going to take the sequence of the nucleic acid, which has originated from a DNA that you inherited, been copied into an RNA, transported into the cytoplasm, and it's going to be translated. If the protein is a glycoprotein, where will that translation occur? glycoproteins are going to be translated by the ribosomes attached to the rough ER. Remember, it has to be in the rough ER because there's no ribosomes on the smooth. And there's differences between free ribosomes and ribosomes that are attached to the ER. So what I'd like to know is what makes a glycoprotein different? Sugars are attached to the polypeptide. Glycoproteins are produced just like any other protein, but when they're produced, they're inserted into the membrane of the rough ER. That insertion allows it to pass into the lumen. That's the space inside of the ER, separate from the cytoplasm. Within the lumen of the ER are enzymes and carbohydrates that can be attached to the polypeptide. If this glycoprotein were to be further modified, in what organelle would this occur? The answer is Golgi. It's going to occur in the Golgi. And you should have some idea of this because proteins that leave the ER are going to move through transport vesicles into the Golgi. This is the next place that they can go to. If they do go to the Golgi, there's additional enzymes within the lumen of the Golgi. Some of these enzymes are going to interact with the sugar molecules, those glucogroups that are on the glycoprotein, and some of them will be modified. So the last question I have is true or false. A glycoprotein from the ER can be transported to and inserted into the plasma membrane or secreted outside of the cell. The answer is true, and here's why. When you look at this picture, you should be able to notice this continuity of the endomembrane system. Information passes from inside of the nucleus through the nuclear membrane via those nuclear pores. That nuclear membrane is continuous with the rough ER, and in fact, the ER membranes in whole. Once it's moved outside of the nucleus and into the information base, it can be translated by those ribosomes and inserted into the lumen. Those transport vesicles, which you see coming out of the ER, are going to be moved and attached to the cis face of the Golgi. Note that the lumen of the ER stays as the center of that transport vesicle. When it attaches to the Golgi and becomes part of the Golgi, it stays within the Golgi. Never do you have this lumen material mixing with the cytoplasm. As it moves through the Golgi and eventually comes out of the trans face of the Golgi, the same is still true. Inside is still inside, and outside is still outside. When that transport vesicle moves to the plasma membrane, that membrane will become a part of the plasma membrane. We're going to study the fluid model of the membrane today, and you're going to see how that sort of comes about. But what's important is that when that membrane becomes a part of our surface plasma membrane, it carries its proteins with it. When it opens up, that lumen never mixes with the cytoplasm and becomes the outside of the cell. Let's take a look at this plasma membrane, or any membrane for that matter, and its composition. You're going to want to pay attention to a couple of different things during our lecture today. Note the phospholipid heads and tails. Those phospholipid heads are going to be hydrophilic, and you can see that they're surrounded by and interacting with water, a lot of water. The phospholipid tails, on the other hand, are going to be separated from the water. they're going to huddle together in water-fearing groups. Note that the water is not spanning through the middle. There's one exception though, and that is a protein. You're going to see this protein more. This is a channel by which water can move through the membrane, a designed selective channel that's made up of proteins. Where this protein is changes those molecular interactions and allows water to move through the channel from one side of the membrane to the other. Now, this complex plasma membrane is the boundary that separates the living cell from its surroundings. It keeps what's inside inside and what's outside out, a lot like those transport vesicles or the other endomembrane organelles. But always remember that it is selectively permeable. Some substances will be allowed to cross it, and some of them will cross more easily than others, which means they'll cross faster than others. And transport proteins are going to be a big part of that controlling of the passage of these molecules across the membrane. Don't forget, when we look at these membranes and the proteins that are embedded in them, there's a variety of different ways in which they can do so. And as you already know, their orientation between each other and within the membrane is going to be important. Here's an animation that just gives you some idea of what might actually be happening in 3D molecular space. This is aquaporin, the same protein that you saw embedded in the membrane that we saw earlier with the arrows pointing to the protein inside of the membrane. You can sort of see here how these molecules of water interact with each other, interact with the surface of the protein, and start moving through the channels. In real life, thousands of molecules of water could move through an aquaporin in every second. So it's going to vastly change those ratios of water and provide control to the cell. You're very familiar by now with the idea that the heads of phospholipids are hydrophilic and the tails are hydrophobic. We call this amphipathic. It quite simply means that there's a difference between one side and the other, a hydrophilic portion and a hydrophobic portion. Other molecules can be amphipathic as well, like those proteins that can span both sections of the phospholipid bilayer. Remember that these are arranged in a very specific fashion, with the hydrophilic heads on the outside, where they can interact with water, and the hydrophobic tails inside. We can show these in a simple schematic like seen here, but in reality, they're moving around quickly, and there's a lot of interaction that's going on, a lot of change that's going on, and a lot of movement. All of this gives rise to the fluid mosaic model. It describes the idea that this membrane that is composed of this fluid layer of phospholipids and proteins within it is not randomly distributed, and it is not set in that distribution. It can change and wiggle over time. This is how, when a phospholipid membrane from, say, a vesicle fuses with the Golgi, those phospholipids can become redistributed in that membrane. It's why when you have the production of membrane from the ER and it travels through various different vesicles, it can replenish the phospholipid bilayer in the membranes of other organelles. Now, phospholipids are the most abundant lipid in the plasma membrane. You've seen that, and you will continue to see that in diagrams. But we're also going to spend time talking about the other components. Membrane proteins are also amphipathic. Lipids can have sugar groups attached to them as well. They don't necessarily have to be just the plain phospholipids we see. As you would imagine, these are called glycolipids. Cholesterols can be inserted into the membrane, more on this in a second, and all of these components stick out into the extracellular matrix. Some of these components, these carbohydrates, have very important roles in cell-to-cell recognition. You may not really think of cells as communicating to one another, but they do so in a chemical way. They recognize cells that are like themselves or different. And a lot of times this is based on the carbohydrates that they have on the surfaces of their cells. One of the best examples of this is how your immune system and the cells of your immune system check other cells in your body to make sure that they match you. For example, a bacteria that invades your body still has a phospholipid bilayer. It can even still have various different and similar proteins on its surface, but your immune system can still tell that it's not you. Membrane carbohydrates are covalently bonded to some of the lipids, which forms these glycolipids. You can also see more commonly are glycoproteins, which is those covalently bonded carbohydrates to protein. Carbohydrates on the extracellular side of the plasma membrane do vary. They vary among species, individuals, and even cell types within an individual. A very common example of the importance of these differences are blood groups. The O, A, B, and AB designations that we give to blood are based on the glycoproteins on the surfaces of red blood cells. Because your immune system recognizes you in part based on these carbohydrates, if you give blood from someone with carbohydrates that don't match you, that can cause rejection. Here's an image of what this sort of looks like. Remember that we have complex extracellular matrices with fibers like collagen and very complex proteoglycans weaving through the surface. These can also interact directly with proteins at the surface and the carbohydrates that are attached to them. All of these are going to be embedded into that fluid membrane. And then some of them will even attach on the cytoplasmic side to some of the microfilaments of the cytoskeleton. Now I have another question for you on your Socrative app. Which amino acid would be most likely present within a transmembrane protein? This is going to be the transmembrane domain, the section that spans across that integral membrane protein, that polypeptide sequence itself embedded within the hydrophobic region. So what kind of amino acid do you think would be found in this center part of the protein? The answer is a hydrophobic amino acid. They give you an example, like valine. This should be an example for you of how like things attract to one another. A hydrophobic amino acid is going to be willing to interact with the hydrophobic tails of the phospholipids, but it's going to be repelled by the watery environment of the extracellular matrix and the cytoplasm. So you might find charged or polar amino acids on the surface or in the cytoplasm of proteins that can span all the way across. They're capable of interacting with the water and are hydrophilic, but hydrophobic amino acids would be found in the center. Now, this fluidity of the membranes can be shown in a very simple experiment. If you take a mouse cell and label its membrane, and a human cell and label its membrane, and then ask the two cells to join together, kind of like the reverse of cell division, what happens is, if you look at those labeled membranes, you find that they've redistributed. Specifically, the proteins have actually rearranged themselves so that they're equally found across the whole membrane. They can't do this without the ability to move through that phospholipid bilayer. Otherwise, you would find all the human proteins on one side and all the mouse proteins on the other. But instead, they've become mixed. This is because membranes are held together by a relatively weak hydrophobic interaction. Not to say that it's not important, and as a whole it isn't very strong, but a protein's only interacting with a certain number of hydrophobic interactions around it. Most of the lipids and some proteins can move sideways within the membrane. That's that rearrangement. And rarely those lipids can even flip-flop across the membrane from one layer to the other. Some different things are going to influence this fluidity, though. In order for membranes to work properly, they have to be fluid. And this is usually about the consistency of solid oil. As temperatures cool, membranes switch from a fluid state to a solid state. In fact, if you were to take olive oil or other sorts of oils that are going to be liquid at room temperature and put them in the fridge, you would probably see that they would also turn solid. As what happens is that the temperature at which the membrane solidifies depends on the type of lipids. You've seen this already with the populations of unsaturated fatty acids and saturated fatty acids. Remember, saturated fatty acids and unsaturated fatty acids have different sources. Those that are unsaturated are more fluid, and so membranes with more unsaturated fatty acids will be more fluid at room temperature. They have to be colder before they get solid than those that are really rich in saturated fatty acids. The steroid cholesterol can also influence this membrane fluidity in animal cells. We know cholesterol as being bad if you have too much of it, but we do need a certain amount. At warm temperatures, like body temperature, 37 degrees Celsius, cholesterol restrains the movement of phospholipids. At cool temperatures, it maintains fluidity by preventing tightly packing. You can think of this as sort of a fluidity buffer. Now, plants do this also, but they have slightly different steroids. Remember, cholesterol is one of these. And so there's different ways to achieve this effect, but they all work similarly. If you look at the fluid state of a phospholipid membrane, you can see all those little unsaturated tails touching each other and taking up space. This keeps the membrane fluid because there's a lot of space for those phospholipids to move around. But the more tightly packed they are, like the viscous model, you can see that there's no longer space. They're more arranged, almost like what you think of when you think of a crystal structure for a solid molecule. Cholesterol fills and creates some of those spaces. It forces these phospholipids to have space between them so that they can move around in certain temperatures. But it also prevents them from having a whole lot of tails and too much space by filling some of that space. Because there are differences in fluidity and there's differences in the temperatures at which organisms live, a lot of organisms have evolved very special lipid compositions to be able to adapt to specific environments. For example, archaea have not only different lipids, but also different linkages that allow their membranes to survive at even very, very hot temperatures and stay fluid when they do so. Fish that live in cold waters have more unsaturated phospholipids. It's one of the reasons why fish is healthier for us than other animal protein. Cold-tolerant plants, like winter wheat, can change their lipid composition depending on the temperature that they're growing at. They're not even necessarily stuck in one configuration. Now we're going to talk about some membrane proteins. We know that the phospholipids form the fabric of the membrane, but it's really the proteins that provide the membrane's function. Peripheral proteins are stuck to the surface of the membrane, primarily on the cytoplasmic side. They're not embedded in the membrane, but they are found at the membrane. And integral proteins actually penetrate into that core. We also call these integrants, such as those that can interact with the extracellular matrix, which we saw last class. Integral proteins that span the whole membrane are called transmembrane proteins. The hydrophobic regions of an integral protein consist of one or more stretches of nonpolar amino acids, and a lot of times these form alpha helices, which you'll sort of see. Note that the protein has what's called an N-terminus and a C-terminus. That N-terminus stands for the amino, and the C-terminus stands for carboxyl. You know now the structure of an amino acid. Each amino acid has an amino side and a carboxyl side, which is what these designate for that growing chain. Cell surface membranes can carry out several functions, including transport, enzymatic activity, cell-to-cell recognition, intracellular joining, and that attachment. Let's take a look. In figure A, the protein is going to be capable of transporting from the inside out or the outside in. And we're going to spend a lot of time discussing the differences between transport in and out. There's also enzymatic activity. So this would be examples of proteins embedded in the membranes of mitochondria or chloroplast, but they can also have membranes embedded into the plasma membrane as well. Signal molecules can interact with the outside of the protein, of the cell, and then actually cause changes on the inside of the cell, like communicating to its surroundings. Cell-to-cell interactions, as you know, involve those sugar groups, those glycoproteins or glycolipids. Intracellular joining is still going to be cell-to-cell interactions, but not based on specificity or this noting a specific cell. These are just going to be cell-to-cell joining. And then, of course, that interaction with the cytoskeleton and the extracellular matrix, keeping together the inside and the outside. So now take a second and use your terminology from this class and your reading comprehension skills to answer this question. Which best describes a biological membrane? If you need more time, pause the video here. The answer is C, 2 layers of phospholipids, with proteins either spanning the layers or on the surface of the layers. You should know by now that there are 2 layers of phospholipids, not a mixture of covalently linked phospholipids and proteins. The layers are very important to function. Proteins can either span the layers or sit on the surface of the layers, like integral proteins or peripheral proteins. You're not going to find proteins embedded between the layers, and you're not going to find proteins freely moving between. Remember, those hydrophilic and hydrophobic interactions determine where that protein is going to be in the membrane. The last one, with 2 layers with opposite orientations of the phospholipids, with each layer covered on the outside with proteins, doesn't account for the fact that proteins span through. I also want to mention that these cell surface proteins are very important in the medical field. We've already talked about how these cell surface proteins determine how your immune system interacts with the rest of the body and with foreign invaders. But foreign invaders can also use these proteins to their advantage. For example, HIV can bind to this cell surface receptor, a protein named CD4. This receptor is going to be used by the protein to stick to your cells. CCR5, which is another receptor, we call it a co-receptor because it helps to actually allow the HIV virus to attach to the cell and then open up the plasma membrane and insert its own genetic material. A recent discovery has shown that some people are actually resistant to HIV. In part, their resistance is due to the fact that the CCR5 that co-receptor that helps the HIV to actually enter your cell is broken. The whole receptor isn't there, and it can't be used by the virus. Because of this natural biological event, scientists are trying to discover ways to be able to block that CCR5 protein, which means that the virus won't be able to find it, won't be able to enter it, and the goal is to stop the transmission of HIV. Now, membranes have distinct inside and outside faces. We've sort of talked about how the fact that lumen stays lumen and the cytoplasmic stays cytoplasmic. Well, you can think about the fact that this would be true for the two layers of the membrane as well. There's an asymmetrical distribution of those proteins, lipids, and carbohydrates in the plasma membrane because this is determined when this membrane is built by the ER and distributed by the Golgi apparatus. So we're going to take a look at this figure, and I want you to make sure that you're looking at the lumen of the ER, how that becomes the lumen of the Golgi, and eventually that extracellular surface. But also keep an eye on what this means for the proteins, the modifications, and their orientation. There's two sides to this lipid bilayer. So we're going to show the lumen side as blue and the cytoplasmic side as pink. Within the lumen of the ER, you have the production of proteins. Some of those proteins can be transmembrane, which means they're actually going to span from one side of the membrane to the other. Others may fully insert into the ER. They may not have any membrane attaching potential. Once these proteins, some of which get glycosylated, get into transport vesicles, they can move into the Golgi. Some of them may go to other places, but those transport vesicles are going to be able to transport them to someplace new. And as we know, the Golgi is a fantastic place to sort and ship things. The proteins that are not embedded into the membrane are still going to be floating around in the lumen of the Golgi. Those that are embedded into the membrane are going to have the same orientation. Transmembrane face pointing the lumen will still be pointing the lumen. This becomes true even as it enters into a vesicle that can take it all the way to the surface of the cell. Note that protein, which is not attached to the membrane, actually leaves the cell. There's nothing anchoring it in place. We call these secreted proteins. Other proteins are going to be embedded into the membrane, like a transmembrane glycoprotein. They are anchored and will stay, but that lumen-facing portion will now face extracellularly. So, true or false? A glycoprotein's sugar modifications can be found in the cytoplasm of cells. The answer is false. Because the sugar modification occurs in the lumen of the ER, and the lumen of the ER will stay the lumen of the Golgi and will eventually become the outside of the cell, there are no glycomodifications found in the cytoplasm of cells. We're going to keep watching in the next video, Lecture 6.2. So please keep your Socrative app running.